what was the reason to express this neuroplasticity. There are the cellular mechanisms at the cellular level, also the modification of cells, of neuronal cells, and at the level of molecular mechanism, and the level of the molecular functioning of the neuron. Well, so I ask you to give me a couple of examples. There are more, many more, just a couple of examples of neuroplasticity at the cellular level, and a couple of examples at the molecular level. You have an idea?
There's synapses, connections.
Yes, synapses. This is just a characteristic of the brain in any case. In neuroplasticity, normal function, in neurodegeneration, etc. But what is the change in the synapses, for instance, under the stimulation of the functional activity of the brain, of the neurons? How it is realized at the level of the cells, of the neurons, at the level of the processes, at the level of the cell bodies, etc. Yeah, please, you are welcome.
If I understood correctly from the last lecture, basically when one part of the brain starts functioning normally, a different area of the brain starts to take over the old function, and the connections kind of generate better. So that's from some, no. That's something I'm taking.
As I understood well, so that's a good idea. Well, so if there is a lesion on the brain, of the part of the brain, then another part of the brain would compensate. You mean yes? Okay. But at the cellular, at the level of the individual cells, what are the functional, what is the manifestation of neuroplasticity? First.
Well, you told us about the spines of dendrites.
Exactly. Spines. Well, so the spines, they correspond to synapses, yes? This is a postsynaptic region of the synapse. Right? And the number of synapses and the spines increased, right? Increased under the functional stimulation of the brain. Good. And then, do you remember what is that stem cells which produce, which give rise to neurons?
And it's a molecular.
No, this is the cellular, because there is a genesis of new neurons, the whole cells, not at the molecular levels. But do you remember what does it mean stem cells, and what is the location of stem cells which give rise to neurons in adulthood in the brain? You're still asleep. Near lateral ventricle, in the stratum, and in hippocampus. Please awake. Okay. Therefore, this is a brain woman. Okay. Well, sure. So that is. Let's see a couple of examples. Well, there are many more. I hope you will recall everything that you examined. And then at the molecular level, what are the mechanisms of molecular mechanisms of neuroplasticity? At the level of the individual neurons. Yeah, please.
I'm not sure, but you were telling us that sometimes neurotransmitters not affect the synapses, but affect body. So as a result, is changing the intensity of producing neurotransmitters.
That's true, but that's not a neuroplasticity. There are two types of interneuronal communication: synaptic context, local synaptic context, and how it is called? Volume neurotransmission, exactly. But that's this synaptic organization certainly is changed under the functional stimulation or degeneration of the neurons. But what are exactly the molecular mechanisms? Look, if the neurons produce neuropeptides as neurotransmitters, and there is a local degeneration of dopaminergic neurons. Monoamine, what happens?
There are monoenzymatic neurons.
Exactly. So peptidagic neurons, they recall that they historically, evolutionary, they were able to synthesize dopamine, and they begin to express enzymes of dopamine synthesis. That's the first one. I said a couple of mechanisms. In other words, the Romania, you have forgotten everything. The next one.
When there is not enough neurotransmitter, for example,
Yeah.
dopamine,
Yeah.
more sensitivity. So I forgot.
Yeah. The
More
sensitivity
receptors.
of the target neurons is increased,
Yeah, and more
You
receptors are appearing.
And what is the molecular mechanism? This is a physiological event, that's under the deficiency of neurotransmitter, dopamine, for instance. There is an upregulation and an increase of the sensitivity to dopamine or to another neurotransmitter of the target neurons. But at the molecular level, what happens? The sensitivity of the neurons, it is characterized by what molecular mechanism?
By amount of receptors?
Sorry, but
By amount of receptors.
yeah, exactly, receptor molecules.
Yeah.
And therefore, the increase of the sensitivity, it is due to the increase of the number of the receptors which are included into the plasma membrane. Okay, good. Nothing? Nothing, remember? Okay, good. Now let's continue.
Excuse me. I
Sure.
have a question about neuroplasticity. And I would like to know that, as far as I understood, those types about which you have mentioned are about the replacement the neurons which were damaged by the neurodegenerative process.
Degeneration, yeah.
Yes, and for such diseases. But if there is any differences in neuroplasticity processes after, for example, traumatic brain damages or strokes.
Well, so as I understood what you said, that there is under degeneration of the neurons in acute diseases, because trauma, this is an acute disease, and there are chronic diseases like neurodegenerative diseases, Parkinson's disease, Alzheimer's disease, etc. Well, so and the neurons, the stem cells, hypothetically, they should replace these neurons, but still there are no good evidence of that. But what was exactly? If it is a response or not?
No, if there is any differences in processes of neuroplasticity after, for example, stroke, because after stroke,
And
some areas of brain which were damaged, the functions of areas which were damaged could be replaced by these areas could be replaced by, for example, the homologous in other hemispheres, and the function will still be functioning after some dimension stroke. And it's also neuroplasticity. Is there any differences from which you have described?
do we mean that if there is a region specificity of neuroplasticity, brain region specificity of neuroplasticity, certainly it is. It is, and I think that the most important neuroplasticity is a characteristic of the most ancient regions of the brain. For instance, basal ganglia. Okay, good. Okay, let's continue our presentation. And the first topic will be hypothalamus as a conduct of neuronal and neuroendocrine regulations. I hope you remember that during our last seminar there was an excellent presentation on this topic. I will, to some extent, I will repeat this presentation, but mostly in the historical aspect, because I was a witness of the history of the development of this neuroendocrinology at my age. Well,
Maybe you should just click on the presentation. Yeah.
Right now.
Yeah, okay. Okay. Well, so look, please. I showed you already. This is a general schematic representation of the role of the brain in the regulation of all the functions, brain functions, and the functions of peripheral organs. Okay, and there is a nervous system which provides a fast and short-term regulation, and neuroendocrine regulation, slow and tonic. And brain is a conductor of both regulations. And now we will turn our attention to the neuroendocrine, to the role of the brain and to the neuroendocrine regulations. It means, look please here, the brain, in particular the hypothalamic region, is a conductor of neuroendocrine regulation. It is responsible for the regulation of all peripheral endocrine cells and also of peripheral organs. And now we will consider the history of the development of this neuroendocrinology. Well, so actually, nowadays we know that there is that the neuroendocrine system is a very complex and very complicated, sophisticated cybernetic system, in fact, which consists of at least of 5 levels: brain and hypothalamus, hypophysis, endocrine glands, and peripheral target organs. Well, and this, according to the feedback regulations, these all 5 levels, they are connected by direct and feedback regulations. Most of feedback regulations are negative, but there are only 2 examples in the whole organism of positive feedback regulation. I will not fix attention on that. Well, so we will turn our attention in this lecture primarily to the hypothalamus and to its role in the regulation of the peripheral functions. Well, so in fact, this is the first topic of our presentation. Well, I will tell you just a few words about the history of the development of neuroendocrinology. And neuroendocrinology was founded, in fact, at the beginning of the previous century, and that was made by the discovery of secretory neurons in the basal region of the brain of fishes, teleost fishes. Okay, and well, so Ernst Scharer, who discovered these neurons, he called them secretory neurons. He considered that there is a special population of secretory neurons. These neurons were characterized by large vacuoles, and he considered that these vacuoles contain hormonal products. Well, do you have an idea how old was Scharer when he made this discovery and founded a new region, a new area in the neuroscience? What was his age?
65 maybe.
How much?
65.
65? Well, not really.
25.
23. 22. That was his, let's say, today we say magister diploma. Well, so he was a very young, very active, and after the World War, he was Jewish, and after the World War, before the World War, he immigrated to the United States. But there were several sources for funding of ideas, of observations, for founding neuroendocrinology. And the next one was made by Hans Selye. Do you know something about this person? That was Canadian. And he formulated the concept of stress and non-specific general adaptation syndrome. Well, so, and what do you think about his age?
30,
How much?
30
Not really.
people.
24, 25. Well, so your Nobel Prize as a kid. Well, and when he was a medical student, and at that time, and even nowadays, all the physicians, they fix their attention to specific symptoms of the diseases. Yeah, you agree with that, huh? But first, he fixed his attention on non-specific manifestations of most diseases. For instance, the increase of the temperature, huh? The headache, etc., etc. Well, so he considered that in addition to specific pathology of the brain and other organs, there is a general pathology which is characteristic of most diseases. And this is a sort of background of the development of any disease. And therefore, he expressed this concept of non-specific adaptation syndrome. He considered this manifestation as the reaction of adaptation. But at this time, he considered only endocrine glands, because he has demonstrated that the axis which is responsible for the stress reaction corresponds to hypophysis, ACTH, adrenocorticotropic hormone, okay, which stimulates the secretion of adrenal hormones, glucocorticoids and many other corticoids, okay? But he suggested that this is a background for the stress. But he suggested there should be a conductor of this local axis. And he considered that this is the brain and mainly hypothalamus. I was happy to, probably I told you, I don't remember, that I was a student in the late of 60s in Moscow. And at that time, we invited, the Academy of Sciences invited many excellent researchers, like Sevier, like Hodgkin. You know Hodgkin? That's a Nobel Prize for the death impulse. Hodgkin, Huxley, Katz. And he was president of the Royal Society in Great Britain. And these people of this level, they were invited to Russia to give lectures, to give seminars, mainly in the Academy of Sciences, in the former Academy of Sciences, but also they gave lectures for public lectures. Well, that was fantastic, because all the students of the university, of medical schools, they were coming to listen to these lectures, and this was fantastic. Really, there was no place even to stay, and it was really. I've never seen the same after all, particularly nowadays. Well, and therefore I was contaminated at that time. I was contaminated by the ideas of Sevier, of neuroendocrinology. And therefore I decided at that time to be a neuroendocrinologist. And I followed this idea. Well, sorry. And there was another group of people, that was Green and Harris. They also suggested that there should be hypothalamic regulation of the hypophyseal function. Because they were pathologists, both were pathologists, and they detected a special short-term blood circulation which was located between the hypothalamus and the hypophysis. And they considered that this blood circulation should serve to transfer signal molecules from one organ to another. But at that time they were wrong, huh? Because they considered that the hypophyseal hormones are transferred to the hypothalamus. But in fact, this is on the contrary. Huh? The hypothalamic hormones are transported to the hypophysis. Okay. Well, so these great people, they founded neuroendocrinology. But that was even in the 19th century, but in the 20th century. But in fact, there were no good techniques to obtain direct proofs of these revelations. And therefore, the most active period of the development of neuroendocrinology, it started from the 60s of the 20th century. Well, at first, that was recognized that these cells, secretory cells, which were observed by Ernst Scharer in fishes, they are also contained in the hypothalamus of mammals here in rats. Huh? Well, so they're located in the anterior hypothalamus, in the very anterior hypothalamus, and they were called magnocellular because they were much larger in size than other neurons in the brain. Well, so these people they considered that these hormones, this should be vasopressin and oxytocin, which are released from their axons in the posterior lobe, having no blood-brain barrier, and therefore they are discharged into the general circulation, and they provide the action on the target peripheral organs like kidney, like uterus, like blood vessels with smooth muscle cells. Well, so these people, they received Nobel Prize in the 50s for this discovery. And then, well, so at that time, people, they considered that all neurosecretory cells are located only in this region. And all of them produce either vasopressin or oxytocin. But there was a very, in the middle of 70s, there was a very great school in neuroendocrinology in Hungary. In Hungary, and it was represented that the most important persons were academicians, Béla Halas, and Béla Flerko in Hungary. And they discovered small cells, small neurons in the middle hypothalamus, green, which send their axons to the hypophyseal port of circulation. And they suggested, there were no evidence at all, but they have suggested that these neurons produce so-called adenohypophysiotropic substances. All these substances, regulatory molecules, which control the functional activity of adenohypophysis. Do you recognize this guy? Nobody? That's me, said this. Okay, good. Now it's, well, so, and finally, Guillemot, that was the French who immigrated to the United States and Chile. They extracted from the hypothalamic region gonadotropin releasing hormone. That was the first discovery of neurohormones in hypothalamic region, and they received Nobel Prize for that. Well, and a little bit later, it has been demonstrated that in addition to the so-called magnocellular center, peptidagic center, and parvocellular, smaller cells, parvocellular centers, both centers are under the control mostly by monoaminergic neurons, which are located in the brainstem. These neurons are represented by mostly by norepinephrinergic neurons and serotoninergic neurons, and they control the functional activity of these peptide neurons. That was the third stage of the development of neuroendocrinology. And nowadays we have a very complicated, very sophisticated picture of the control, of the hypothalamic control of neuroendocrine regulations, with direct regulations by neurohormones, which are released either into the hypophyseal portal circulation or to the general circulation, by their control by monoaminergic neurons, and by the feedback control by the hypophyseal hormones of peptide-ergic neurons. Is it clear? Yeah, please.
It's not there with the feedback. How about the feedback system?
Feedback. This one, yeah? Feedback. Well, so look, these hormones, pituitary hormones, they cannot penetrate the blood-brain barrier. You're right, huh? Therefore, theoretically, they cannot be released to any brain region. But they can enter the brain via so-called local regions, and they are called, how they are called? Small brain regions which lack circumventricular organs. Good. Well, and in the hypothalamic region, there are two circumventricular organs. Two, right? One is the median eminence. In this region, look, the third ventricle is very close to the hypophyseal portal circulation, which is fenestrated. And there is no blood-brain barrier, and actually there is almost no barrier between the cerebrospinal fluid and the hypophyseal portal circulation. Okay? And there is another one, circumventricular organ in the hypothalamic region, in the very rostral region. Yeah, it is not drawn here. It is called organum vasculosum of the lamina terminalis. Well, so there are two organum vasculosum of the lamina terminalis. That's a fun story. Well, okay. That's clear? I'm not too fast, no?
Can you repeat the organum?
Organum vasculosum of the lamina terminalis in the very rostral region of the hypothalamus, and in this region mostly the gonadotropin releasing hormone axons are terminated. Well, so organum vasculosum of the lamina. And how many circumventricular organs in general in the brain? How many? In the hypothalamic region too. And how many more? In other brain regions.
I have seen subphonical.
How many?
I have seen subphonical area. I don't know.
Yeah. How many? How many more?
Four.
I have the number 7.
7.
There are 7 in total.
In general. Yeah? 7. Other ideas? 5.
5.
5. 3 in addition. Subcommissural, epiphysis, etc. What's 3 additional? And what is the characteristic of the anatomical distribution of this circumventricular organ in the brain? Do you remember? What is characteristic of the distribution? There is a specific characteristic of the topography in the brain. All of them are located in the mid-sagittal plane of the brain. You understand what this means? Mid-sagittal plane. Okay, good. Well, and now, that's mine. Well, I just returned again to this idea of the circumventricular organs, and you can see all 5 circumventricular organs in the brain. Subcortical, subcommissural, organomusculoso volante, organomusculoso volante terminalis, media neminis. Posterior lobe that is not supraventricular organs because there is no contact with the cerebrospinal fluid, only with the blood. And well, and now let's go. Well, and you can see that at the level of these supraventricular organs, there are all very sophisticated, very complex relations between the neurons, secretory neurons, and cerebrospinal fluid, the ventricle, and blood vessels. Look at it first. Well, so there is no blood-brain barrier, right? The vessels are fenestrated and easily pumpable to any substance. Then, the neurons, the secretory neurons, send the axons to the cerebrospinal fluid, and they discharge the secretory product into the cerebrospinal fluid. But at the same time, the same neurons, they send the axons to the blood vessels, and they discharge the secretory product in the vicinity of the blood vessels. Well, and therefore, in this region, there is no blood-brain barrier between the brain and the blood circulation, and there is no barrier also between the ventricular system and cerebrospinal fluid and the blood vessels and bloodstream. Okay, and look, please, how is the complex regulation of the release of new hormones from the axonal terminals. Look, it is under the control at two levels. First, at the level of cell body, there is a control by other neurons. And these neurons control two functions of the neuron. First, the synthesis of these neurohormones. And second, the release of these neurohormones from axonal terminals. And also there is a regulation at the level of the axonal terminals. And there is only one regulation of one function. This is the release of neurohormones. Okay? Then there is another level of the regulation, neuroregulation, called blood vessels. Well, so these neurohormones could control the penetration of substances. Even if there is no blood-brain barrier, there is a regulation of the transfer of substances through the epithelial cells. And finally, there is also a regulation, direct regulation, neuronal regulation of hypophyseal secretory cells. Also, there is a sort of synapses, not real synapses, because these are epithelial glandular cells, but this sort of synapses, and the neurons, they control secretory activity of hypophyseal secretory cells. Well, so look, please, how is it sophisticated, complicated regulation? First, at the level of cell bodies, then at the level of axonal terminals, then at the level of blood vessels, and finally at the level of hypophysis. Well, so this is, in fact, this is a very complicated system of adaptation, because this system is responsible for the regulation of most important functions. It should be very flexible in its functioning, particularly in pathology or in extreme situations, stressful situations, etc. Well, let's go then. Well, so it is shown by rows the release of different substances for different places. Well, now let's turn to the second point. It says circadian timing of the brain function is an adaptive mechanism. Well, you know certainly that there is a pacemaker in the heart. You know, everybody knows, yes? Yes. Pacemaker in the heart, which controls the activity of the heart. But this is the pacemaker which is functioning only for the heart. But there is a brain regions which is responsible for the circadian rhythmic activity of all the functions in the body and in the brain also. How it is called, this region?
Is it the suprachiasmatic nerve?
Suprachiasmatic nucleus. Good. Next time I will ask you to give a lecture. Well, it would replace me. Well, and well, so, this is suprachiasmatic nucleus. Look, please. It is called biological clock. This is biological clock, not only of the hypothalamus, but of the whole body, directly or via the intermediate links. Now, please, this very small region is located in the ventral portion of the hypothalamus. This is the third ventricle. This is the optic chiasma. And these are very, very small neurons. The smallest, probably, in the hypothalamic region. And they consist of a number of peptidic neurons, which produce vasopressin, vasoactive intestinal polypeptide, the somatostatin, and some others. And what is important? That these neurons are absolutely different from all other neurons in the brain. They function according to their circadian genetic program. Well, so if we this genetic program with the period, with a cycle of 22-24 hours, okay? How it is proved? If we take this, if we separate these neurons from the suprachiasmatic nucleus, good morning. Very nice. Well, if we separate these neurons, if we descend these neurons from the suprachiasmatic nucleus and put them into another brain region or in cell culture, dissociate, they would continue to generate the same cycle, circadian cycle of the synthesis of these peptides, sort of messenger RNA and their peptides. Well, and but still, so the circadian rhythm, on the one hand, they are genetically predetermined, but on the other hand, there is a small little control, maybe little control, little bit modification, but by 3 neuronal inputs, efferents. This nucleus is innervated by serotonin-energetic neurons of the rougher nucleus, by neuropeptide Y-producing neurons of the gingulated leaflet, and also by the axons, which are coming from via optic nerve. Okay, so there are 3 sources of afferent regulation of suprachiasmatic nucleus. And well, but this regulation is not really crucial. Now let's go. Well, so then this is a schematic representation of the pacemaker output, because these neurons of the suprachiasmatic nucleus, they project their actions to different brain regions, and thereby, and by this way, they control the activity of these neurons due to circadian rhythmic activity. Well, so there are axonal projections, the thalamus, probrain, paraventricular hypothalamic neurons, preoptic hypothalamic region, tuberal, and almost to all other brain regions. And now let's come to the conclusions. Circadian rhythm is an adaptation mechanism of living organism to the environment. Well, so that's a good example. Well, in December, I spent my vacations for 2 weeks vacations in Cuba, and there is a difference for 8 hours in time. And after coming back, I couldn't sleep overnight, at least for 1 month. Normally, asleep. Well, then, circadian function is controlled genetically by the expression of genes that encode for specific proteins. Huh? You remember that there is a genetic program which is functional in situ and also in vitro. Okay? Neuronal circadian timing system establishes the temporal organization of behavior into cycles. Well, so there are general conclusions. And I repeat once again that it's fantastic that the smallest nucleus in the brain control all the functions of the body and of the brain according to the circadian rhythmic activity. And now let's turn to the general conclusions, to the hypothalamus and to the circadian rhythmic activity. Brain is a principal conductor of the function of the whole body due to the neural and neuroendocrine regulation. The hypothalamus is an integrative center essential for surviving and reproduction. Well, so do you remember why I fixed your attention on surviving and reproduction? Do you remember?
Because it's the main
That's-
functions of the brain.
Sorry?
Because it's the main functions of the brain.
No. There are two most important functions of organism, of bodies. This is reproduction and survival. Surviving due to the adaptation to the changes in the environment. If the body cannot explore these functions, they cannot survive. Right? The organism cannot survive. Well, so therefore I fix your attention to this adaptation to the environment and reproduction. But certainly not only, it controls not only these functions. Somatic organs provide the chemical link between the brain and periphery. Neuroendocrine system is a multi-level cybernetic system based on direct and feedback regulation and solving domain homeostasis. Could you tell me what are the levels, structural levels of this cybernetic system which are linked by direct and feedback regulation? Do you remember? There are 5. Huh? Huh? The first one? Huh? The brain. Huh?
I just don't really think you kind of paid enough time, to list all the 5 levels. No?
Sorry.
Oh, okay.
No, but probably that's a problem of the language. What do you mean? Could you explain this?
I just don't think that you specifically pointed out all of the 5 levels.
Yeah.
So,
Yeah, I pointed.
yes.
I showed them.
Okay, maybe
I showed
just
it.
wasn't-
There was a very, I showed you that was in blue. Huh?
Yes.
That was a very complex neuroendocrine system which functions like the cybernetic system with 5 levels, structural levels, which are connected by direct and feedback regulations. Nobody remember what are the levels? The first one, this is the brain. You should switch it on. Huh? Here the lectures. Well, then the next one, the brain and the hypothalamus, yes? The next one, that is
So,
hypothesis. The third one,
second one is hypothesis.
this is endocrine glands. And the last one, help me keep that. The last one, the peripheral organ, target peripheral organs. Okay?
This can you go by again?
Sorry?
The levels can you?
Yes, can you name them again?
The
So I
level
think you
five.
only named 4.
What is the buff level?
The brain.
The brain is the first one. And the brain it receives all the information from so-called inter and extra receptors. What they mean? From the receptors inside the body, from the blood, etc., and on the skin, for instance, etc., and external. Okay, good?
You should please repeat again because I think you said 4.
Yes, you only need
For brain,
Probably when saying 5, I mean the brain and hypothalamus separately. Yeah? Okay, look, brain, hypothalamus, hypophysis, endocrine glands, and peripheral organs. Okay? Well, so in fact, you are right because we could consider brain and hypothalamus as the only one level. Okay, good. Well, and in addition to neuroendocrine regulation, circadian timing of the brain function is a crucial adaptive mechanism. Superchiasmatic, and there is a really adaptive mechanism because over this month, when I could not sleep after Cuba, it was extremely difficult for me to work the whole day after all. Well, superchiasmatic nucleus is a pacemaker of circadian rhythmic activity of most functional systems. It's clear?
I have a question. It's clear, but is there any opportunity for neuroplasticity here? I mean, after the brain damage for hypothalamus or any of this aforementioned organs, is there any opportunity that some other brain region will take this functions?
Well, so if we're talking about superchiasmatic nucleus, certainly no. Certainly no, because this is a completely unique structure with unique neurons. So, well, as for secretory cells in the hypothalamic region, which produce the hypophysis-entropic hormones, well, there might be mechanism for neuroplasticity. First, this is the activation of some, well, so if there is a damage of some neurons, there is an increased activation, there is an activation of others, functional activation, on the one hand. On the other hand, I could, I don't know exactly, but I could imagine. Well, so these neurons which produced adrenaline hypophysis-entropic peptides, hormones, they are located not only in the hypothalamic region, but they are located also in other brain regions. And in other brain regions, they are responsible for the control of other functions, of memory, for instance, vasopressin, or memory, gonadotropin releasing hormone for sexual behavior, etc. But I could not exclude, probably this is the case, that these neurons begin to secrete the peptides into the general circulation, while the supraventricular organs. It's really logic, but I couldn't give you any direct reference, because it's quite logic. It should be. Okay? Good? Okay, let's turn further. That development, now we'll turn to the development of the brain, regulation of brain development, and neuron differentiation. But so I would like to emphasize that it should not be thinking that the brain, the developing brain, is the same as in adulthood, but only smaller, okay? Because there are quite fundamental differences between the functioning of the brain over the development and this regulation. Do you remember? I told you probably 3 times that. What is the difference between the action of the same chemical signals, physiologically active chemical signals, on the same receptors of the target neurons in the brain in adulthood and during the development of the brain? Do you remember what is the difference? The same signals which affect the same receptors in the developing brain and in the developed brain, just a moment, but the action, final action, is fundamentally different. Please, Yolanda.
If I remember correctly, I think you stated that in the developing brain, there is a more long-term effect.
So, absolutely. Absolutely, because this is absolutely different action. Because in adulthood, this is a short-term action, only during the period of association of chemical signals with the receptor molecules. But this association, it lasts for seconds, no more, for seconds, and then this functional action disappears. But in the developing brain, the same chemical affect the same receptors also for a relatively short time. It could be for seconds, for minutes, etc. But its action is manifested over the whole life. This is a so-called morphogenetic action. Well, so this is an action which provides a control of the development of the brain, not of its functioning, but of the development. Clear? Huh?
Yes, and can you please clarify until what age does this effect happen?
Okay, good. Well, so we will consider this point a little bit later in more detail, but I could tell you that this is a period, usually the period, so-called perinatal period. This is the end of prenatal life. From the end of the prenatal life, or the last third of the prenatal life, and the early postnatal life, which we call neonatal life. In humans, I think this is the period. Well, so in humans, you know that there is a short period, you know better than me, that the period of pregnancy is 39 weeks. Well, so this action is started shortly after the appearance of the neurons. This is in the last half of pregnancy, and it lasted at least until the age 5-8 years after that. But I will show that a little bit later. Good. Let's go on. Good. Well, so development of the brain and differentiation of the brain neurons. The first point. So remember there was a presentation at our seminar showing that the brain is developed, is manifested as a number of swellings or bubbles. We call brain bubbles or brain swellings. Initially, this is a swelling for 3 bubbles, 1, 2, 3, and then for 5. And the neurons and the stem cells, which form the wall of these ventricles, they give rise to neurons. These stem cells, that are intensely proliferated cells, but after the seizure of their proliferation, they give rise to neurons. Well, so the neurons, they're produced by the cells of the wall of the ventricular system. Okay? And after that, they migrate to the places of their final settling in the brain, where they provide their specific control. Well, so when we summarize, there is a lot of studies on the development of the brain, and I schematically summarize this data. First, as I already mentioned, that the neurons are generated from matrix cells or stem cells, which are intensely proliferated. And after seizure of this proliferation, the neurons appear, because neurons, they do not proliferate. Understand? Well, so this is the first step, neurogenesis. Neurogenesis. After all, there is a migration of these neurons from the wall of the ventricles toward the places of their final settling and of their functioning, like hypothalamus, like basal ganglia, like cortex, etc. Okay? Well, then the third stage of the development of the brain, this is establishment of neuronal networks via synaptogenesis. Well, so the neurons establish the specific context, like this, affirmative generation, with the establishment, with the development of synapses, synaptogenesis. And finally, I repeatedly mentioned that some cells, some neurons, they are not capable to find out their targets, and therefore they die. They have no job, and they die. And therefore, in prenatal period and in the early neonatal period, there is a great level of apoptosis, of the death of the neurons. The same, very similar to that happens in people, aging people with the neurodegenerative diseases, but at the very early step of the development. Well, and apoptosis, okay? This is a genetic program in a cell death. Yeah, please.
For how long the apoptosis is happening?
The apoptosis of individual neurons or the whole population. Well, so there is a lot of neurons which are passing apoptosis before. These are, I think, thousands of neurons which die before birth and after birth. A lot of neurons. These are not single neurons. There is a lot of neurons. Well, it means that the brain, it produces many more neurons than it really needs. This is also a sort of compensator of adaptive correction.
And at what age this is of the neurons?
What is the age of apoptosis?
When the apoptosis ends?
Yeah. Over the neonatal period, that after birth, I think most in humans, for instance, I think that 1 to 2 years after birth. Okay.
So, I remember hearing about changes
A
in
little bit louder for everybody, because
I remember hearing about some brain changes after a person is 25, I believe. So there is also a gap of neurons having this age.
but there is the period of the development of the brain that is considered. This is a period of the final establishment of synaptic relations and interneuronal relations. And at the same time, there is an establishment of the blood-brain barrier. If we're saying about rodents, this is the end of the first month after birth. If we're saying about people, this is several years after birth, 5-8 years. Well, so there is a modification of the brain all over the life. In aging people, there is a faster degeneration of neurons, etc., etc. But I don't know that there are really some specific age related after these 8 years old. I'm not sure that there is an age-specific local timing, local modification. Yeah,
Yes, maybe you'll touch on the subject a little later in the presentation, but I was curious to know how do neurons exactly how they migrate, because I understand they go from the ventricle areas thanks to chemical signals, but I don't quite understand like the neuron on the picture is wrapped around something, and I
Yes, good question. Look, please. This is an enzyme, okay? This is the basal surface, huh? And this is head region here, okay? Well, this is a layer of matrix cells, okay? The matrix cells, they have a very long processes, basal processes. Okay? And when the neurons appear, are generated within this layer, epididymal layer, they migrate along these processes. These processes plays a role of a sort of a bridge for them. You understand? Well, so this is drawn here. This is a process. This process, okay? And along this process, they migrate to the regions of their final segment. Understand? No?
Can you just clarify on what you've drawn? What is what? Because like I understand that that's a
Look this. It is not drawn here. It is not drawn here. The ventricle, which is drawn here. Okay. It is not drawn there. Then there is an epithelial layer or epidermal layer, which is rich in stem cells. Okay. But it is drawn here. This process. This one. And this is a basal process of this matrix cell, which is projected into different regions of the brain. And therefore, this neuron, which is generated in this epithelial region, it is migrate via this track. Okay?
I understand better now. Thanks.
What exactly are those basal processes? What is
What big?
basal process? What's that?
What's that?
Yes, what is that? Basal process.
I don't know the wrong exactly. Well, so look, the wall of the ventricle is formed by the cells with long basal processes.
Yes, that basal
But-
process.
So this is just more. This is a cell body, okay, and then there is a basal. The only one basal process.
It's like
Axon of the cell.
It's like an axon, but without dendrites.
Okay.
Well, so the cells they can have many processes, but this type of cells they have the only one process.
oh wait, so the word process is an umbrella term for dendrites and axons, like the projections of the neuron. Yes.
No, if we are saying about this process, it serves as a road for migration of neurons which is generated in this region. Well, so it is used as a road, as a railway.
So, Prof, it's a tube, more
Yeah.
like a tube. The stem cell travel through, something like that.
We can say so function, but topographical not those. Not the same. Okay, let's go on.
Yes.
Good. Well, and now let's turn to chronology of the development of the brain in human being. Well, so this is a neuronal proliferation, and in fact, this is a period of the genesis of the neurons, decision of the neuronal of the progenitor cells proliferation. It happens until the 12th week, gestational week. Okay. Then there is a period of neuronal migration. Neuronal migration. This period takes place at least under the birth. Okay? And and then from the 20th prenatal week to the adolescence, I think this is 4 or 5 years old, huh? There is a period of the establishment of synaptic interrelations between the neurons. Okay? Synaptogenesis, which is called synaptogenesis. Huh? Well, and as I mentioned already, that during the perinatal period, there is an apoptosis of the excessive number of the neurons, which are not functionally important for the brain. Good? Apoptosis. And now these projections are very interesting for you because you're a specialist in highest physiological activity and cognitive functions. Look, the maximum of the establishment of sensory system, brain system, that corresponds to 3-4 years after birth. All of them after birth. The language capacity is established, the maximum is 8 years old. And the high cognitive functions, this is months, I'm sure this is not years. Huh? And the cognitive function, the maximum is 3-4 years old. Okay? Do you have this presentation or not yet?
Not here.
I didn't see it. That's right. I will send you today. Unfortunately, Lisa is thick actually, and I will send you. Okay, let's go there. And then the crucial question for the development of the brain, in particular of the neuroendocrine system, which is under the control of the hypothalamus, most of the hypothalamus. This is the development of neuroendocrine regulations in neurogenesis and its functional significance. If this is the same as in adulthood, just to control the functional activity of the target organs, or it is important for the development of the peripheral organs. These are quite different points. Yeah? Well, and before the end of the 80s, there was a concept that was developed by very famous scientists and by my former chief, my supervisor, Professor Mickiewicz, and they contributed something to this concept before the end of the 80s. What does it mean? This concept declared that the brain that has no any role in neuroendocrine regulations until its full maturation, which happens very late in rodents at the end of the first month after birth, and in the humans, I think that several years after birth. You're super charismatic. Nucleus doesn't work much. You should sleep overnight, but not the lecture.
Right.
Sorry. No, that's for you, super charismatic nucleus, not for you. Well, and so that was according to this concept. But on the contrary, over the perinatal period, in embryos and after birth, the development of the brain is under the control by the secretory products of endocrine glands. Well, so it has been suggested that the brain is developed under the morphogenetic control by hormones of the endocrine gland, which are developed very early in ontogenesis. Well, and only the full maturation of the brain, very late in ontogenesis, it begins to control pituitary function as in adulthood, and why pituitary functions, all the endocrine glands. That was the concept which was developed until the end of 80s. it says that the prenatal and neonatal open looped of neuroendocrine. So there is closed looped neuroendocrine gland. It appears only late after birth. Huh? Yes. Yeah. Well, and then I told you many times always that the real discoveries in science, they are made usually if we find some contradictions in the data. Contradictions in the data and raising the provocative questions to ourselves. And I know I discovered for myself that the neurogenesis, it happens in rats. In rats, there's a period of gestation, 23 days. This is the 12th day, this is the first postnatal day after birth, and this is the 15th day. This is adult. Okay, well, so I compared this data which were accumulated until the late 80s. The neurogenesis, the neurons are generated over this period until the 15th, 16th day of prenatal life. But what is interesting, that these neurons, which have nothing to do with neurons, they have no neuronal processes, no axons, no dendrites. They look like balls, small balls. They, over this period, just after the appearance, they begin to express all the specific synthesis. All the synthesis of physiological active substances, all the synthesis of receptors, neuronal receptors, and all the synthesis of transporters. And without being neurons at that time. And only later, at the end of prenatal life, they begin to give rise to processes, axons and dendrites, which outgrowth from the cell body, only at that time. Okay? And only after that, at the late period, 15 days after birth, there is an appearance of the development and the closing of the blood-brain barrier. So, look please. Oh, sorry. Look please, what is interesting? That over this period, there are no really measured well-developed neurons on the one hand. But they secrete all the physiologically active substances. And at the same time, there is no blood-brain barrier. And therefore, these substances, according to my hypothesis, these substances can easily penetrate into the blood circulation and providing a hormonal regulation, endocrine regulation of peripheral organs of the body and also of the brain itself. Endocrine, distant, okay? That is absolutely impossible in adulthood after the appearance of the blood-brain barrier. So in other words, I consider the developing brain functions over this period as a multi-potent giant endocrine gland, endocrine organ. And that was, at that time, that was a paradoxical reality. Nobody believed me. But in order to prove this hypothesis, we should show that the concentration of the brain-derived neurohormones in general circulation before the closing of the blood-brain barrier in ontogenesis should be as large as that in the hypophyseal portal circulation, okay, in adulthood. The next experimental evidence that we needed This is the concentration of the blood-derived neurohormones in peripheral blood should drop under inhibition of their synthesis in the brain. It means that only the brain is a source of these substances in the blood, not peripheral organs or maternal placenta. And the third evidence that we lead that circulating brain-derived neurohormones should contribute to the endocrine regulation of the developing peripheral organs and the brain itself, which we can call auto-regulation. Okay? And now we certainly the brain. Look, in adulthood, there is the most potent, the most efficient endocrine gland. This is hypophysis. It produces 8 hormones, a little bit more or less. 8. But the brain, when functioning as endocrine gland, should produce hundreds of thousands, even thousands, physiologically active substances, which hypothetically could contribute to the regulation or the development of the brain and the whole body. But certainly, I had no time in my life to test all these 1,000 substances. And therefore, we selected only 2 markers of the, of the brain functioning as an endocrine gland. This is dopamine and GnRH, gonadotropin releasing hormones. Why we used this? Because in adulthood, in the peripheral blood, there is no these substances. Because all these substances which are produced in a high amount in the brain, their release is prohibited by the brain-blood barrier. And therefore, in the blood, there is almost 0 of these both substances. And therefore, we have used these substances as the markers, biomarkers of the brain as an endocrine gland. And the first question was whether the concentration of neurohormones in peripheral blood in ontogenesis before the blood-brain barrier closing is sufficient to provide an endocrine control of target neurons. In other words, whether these markers are in a high concentration in the blood vessels, in general circulation, and in this concentration, it is capable to provide its action, physiological action. Well, what we have done? We have inhibited, look, first, we have shown what is the change in the concentration of these substances during ontogenesis, before closing of the blood-brain barrier, and the blood-brain barrier is closing around 15 postnatal day, and after. Look, please, before closing the blood-brain barrier, there is a high concentration of GnRH. There is a high concentration of dopamine. Okay? But after the development of the brain-blood barrier, the concentration dropped of GnRH and of dopamine. It means that that dopamine and GnRH, which are circulated in the bloodstream, it is capable to provide the physiological action on the developing target organs, peripherally in the brain itself. The second question was whether the developing brain is a principal source of circulating neurohormones. You know many examples that during the development, the different organs, peripheral organs, the brain, they express temporal synthesis, transient synthesis of some physiological active substance. And therefore, we could not exclude that this GnRH and dopamine, which were detected in the blood circulation, are synthesized not only in the brain, or not in the brain, but in other peripheral organs. And therefore, in order to prove it, that the source of these substances is the brain, we inhibited the synthesis of these substances in the brain by using stereotaxic techniques and the toxins of these neurons. Okay? And look, please, after inhibiting of GnRH synthesis only in the brain, and dopamine synthesis only in the brain, the concentration in the plasma decreased significantly. Certainly, we did not destroy 400% of the sources in the brain of generation, and therefore there is not a drop to zero. But this is a direct evidence that the generation dopamine, which were found, which were detected in the peripheral circulation, are of the brain origin. This is the second evidence of this hypothesis. And the third one, whether the brain-derived neurohormones provide a direct endocrine action on peripheral targets and the brain itself. Well, so even when looking at the literature at that time, we can see that there is a lot of peripheral organs and the brain regions which are sensitive to generation and dopamine, and also to serotonin and other. Look, the testes are sensitive to generation, kidney to dopamine receptors, etc. Well, so this generation dopamine, which are released from the brain, they have their own targets in the periphery, and they could contribute to their development. And now let me show this is summarizing scheme of this, let's say, discovery. I told you previously that when I was a young scientist, and people at your age, a little bit older, people asked me to write an annotation of my achievements in science, my contribution in science. I wrote 3-4 pages. Now I write 4-5 lines. And this is one of the lines. This is really, I consider this very important discovery. And this is a new field for the development of neuroscience for ages, for decades. Look, what is the general scheme? Well, so at the beginning, there are neurons, but non-differentiated neurons in the brain. They have no processes. They have only cell bodies. But there is already synthesis, expression of all specific synthesis. Physiological active substances, receptors, and transporters. And they release these substances. And it is believed nowadays, that's not my idea, it was shown, that these substances could control the development of the next neurons, providing so-called paracrine regulation. Well, the neurons which are in the vicinity of this neuron, because there should be a short distance for the diffusion of these substances. In fact, the substances which are produced here, they cannot diffuse and to reach these neurons, which are far from the source of this physiological substance. Therefore, these substances could provide only outer regulation or paracrine regulation. Okay? Well, but we emphasize that in this period of development, there is no blood-brain barrier. And therefore, we have demonstrated that these physiological active substances are released into the general circulation. And therefore, they are capable to provide, and they provide, endocrine control, endocrine distant control, on the peripheral organs, over this periphery. Moreover, the substances which are released from this neuron can provide an endocrine regulation on the distant neurons via the feedback. Well, so the substances which are released from these neurons, they cannot diffuse and reach these neurons via the brain, but they can reach these neurons via blood circulation. Well, so there is on the one hand, these substances provide control of the development of peripheral organs, and on the other hand, they provide the regulation of the development of distant neurons in the brain, autoregulation. Then, after closing the blood-brain barrier, there is an establishment of there are outgrowth of the processes, of the axons, the establishment of synaptic interrelations, but since this time, there is no diffusion, there is no release into the general circulation. And now finally, we return again to the concept which was developed before the end of the 80s, and it said that there is an open-looped regulatory system, and the brain has nothing to do with neuroendocrine regulations after its full maturation. But finally, we came to the just opposite hypothesis, and it is proved actually. It's not a hypothesis. This is a theory already. That the brain, over this period, ontogenesis, the brain functions as a multipotent gene endocrine gland, which produces doses of physiological active substances, and it controls the development of the whole body on the one hand, and also of the brain itself via endocrine regulation. Yeah.
I would like you to clarify, so you believe that these substances, when there is no blood-brain barrier, affect only the endocrine system when it's developing, or just generally things outside the brain as a whole?
Now, before the establishment of the blood-brain barrier, these substances are released into the general circulation, and they control the development not only the endocrine glands, but all peripheral organs which express receptors for this chemical signals. For instance, GnRH has receptors not only on the pituitary gland, but also on gonads. And therefore, over the perinatal period, GnRH control the development of the hypophysis and of the gonads. Gonads, these are peripheral organs.
So I guess my bonus question would be, why do you think this would be necessary for our development as a whole? Like these substances, why do they need to be circulated through our bodies as we grow? What is the hypothesis?
You know that that's my feeling. That's my feeling. That to maintain neurotransmission in adulthood in the brain and the endocrine regulation of peripheral organs, my endocrine organs, the body needs only doses of physiological active substances, not a lot. Doses, 20, whatever, but no more than 100. But for the developing organism, we need many more physiological active substances. Some of them we know, but I think this is only a top of iceberg. And therefore, the brain at this period, it is a source of hundreds and thousands of physiological active substances, and we do not know what is the action effect on the development. But I believe that is very important, especially for the regulation of the development. And therefore, I told you that this let's say, discovery, it opens a prospect for the development of this area of neuroscience for decades. Yeah. Yeah. That's the next We should take a break. Okay. You will have for 10 minutes.
No, it's 10 minutes since we should begin the break. The break is for
Well, so we should take a break for right away, yes? For what time? 20 minutes. Okay. Let's see. 7 minutes break. Well, there is no enough concentration, enough high amount of these physiological active substances, or there is no enough sensitivity for these substances of the target neurons. This should result in the development of so-called congenital diseases. Congenital diseases. And these diseases we cannot improve, we cannot treat. There is no way to treat congenital diseases after birth. Therefore, the only way is to develop an early diagnosis during the brain development, intrauterine development, or just after birth, just to compensate the deficiency of some or excess of some physiological active substance. And look, please, that there are several examples of congenital diseases. For instance, Kalman syndrome. I told you, I don't know, during last lecture earlier, the Kalman syndrome, this is a hypogonadism. This is an impaired regulation of reproduction. You remember that GnRH neurons in the hypothalamic region are responsible for the regulation of reproduction by control the expression of luteinizing and follicle-stimulating hormones of hypophysis. Okay. Well, but these neurons should be located in this, concretely in this hypothalamic region, in order to secrete GnRH into the hypophyseal cortex stimulation. In Kalman syndrome, these neurons are generated normally, but outside the brain. In NOM and in syndrome like Kalman syndrome, GnRH neurons are generated in the nasal cranium, not in the brain, not in the hypothalamic region, but in the nasal cranium. And only after that, they migrate along the nerves, cranial nerves, into the brain, into the rostral part of the brain, and then to the hypothalamic region. But in Kalman syndrome, in contrast to normal people, the neurons are well developed, are well generated in the nasal cranium, but they cannot, they have no capacity to migrate to the brain region. Well, so they for the whole life, they are located in the nasal cranium. They function normally, they secrete GnRH, but GnRH is not released into the hypophyseal cortex circulation, and therefore it is not involved in the regulation of reproduction. Well, so it is called Kalman syndrome. Okay? Well, so there is also attention deficit disorder, which is believed that this syndrome is related also to the deficiency of the hormone. There is a heart pathology which depends on the deficiency of serotonin, and precisely at the end of fetal life, there are serotonin receptors on the heart, which are responsible for the development of the heart. If there is no serotonin receptors at the end of prenatal life, the children, the embryos, they cannot survive after birth. Norepinephrine is related to sudden infant death syndrome. It's an autism, serotonin, autism, heart pathology. Well, so there is a lot of congenital diseases which cannot be treated after birth. The mechanism of most of these congenital diseases are not still clearly understood. Well, now let's make a conclusion to the concept on the role of the brain in neuroendocrine regulations in ontogenesis. What was considered, what was believed in the past, what is believed, what is considered at present, and what should be done in the future. Well, so in the past, we consider that the brain does not participate in neuroendocrine regulations until its complete maturation. In rodents before birth, before puberty, but its development is controlled by morphogenetic factors of brain and peripheral origin, mainly by steroid hormones, by sex hormones. What we know at present, that the developing brain operates as a multipotent endocrine gland that provides direct regulation of peripheral target organs and the brain itself via auto-control, auto-regulation, in autogenesis from the birth of neurons to the establishment of synaptic interneuronal relationships and closure of the blood-brain barrier. What should be done in the future? That's mostly for you. Huh? It should be determined what is the role of brain-derived morphogenetic factors in autogenesis before closing the blood-brain barrier in the regulation of functioning of morphogenesis of the target peripheral organs and the brain itself. And what is the role of the entire metabolism of brain-derived morphogenetic factors in the development of congenital diseases? And certainly, according to the paradigm of translational medicine, this data should be translated to the development of new technologies for early diagnosis and treatment of developing diseases. Well, I think that we should just. Well, I think that I should start my lecture, and we will turn to your presentations. And I will first make a small information. Well, so today we have two presentations. Nikolai Kultsov, the nervous system of the brain, and Anton Belkak with the synaptic plasticity and stimulation. And unfortunately, Alice will lose reception because she's sick. And also, and for the next seminar, we have three pages. I will show them, and you will see.